colleagues from ICAR-IIGR DGR, now IIGR. So, good morning, good afternoon, everyone. So, can I share my presentation?
Yes, sir. Yes, sir. There are, sir, any opening remarks? Shall we?
Go ahead.
Go ahead, sir. Okay, okay. Thank you, sir. Thank you.
My presentation is visible?
Yeah.
Full screen?
Yes, sir. Full screen.
Audible now? I'm audible also?
Yes.
Go ahead. You are audible. Yeah. I'm Dr. R.D. Prasad, Principal Scientist, Plant Pathology, and I have been working in this Institute of Oil Seed Research since 2002. Before that, from 1995 to 2001, I worked at Project Directorate of Biocontrol, Bangalore. Now it is NBR, that is Insect Resources now. So then onwards, apart from our ICRI programs and crop improvement and production programs, I have been continuing my work on microbial biological control for various plant diseases. So today I have my this information on these biologicals in seed treatment. So mostly I will be talking about this a little bit of introduction on the subject, as well as the mostly I focus on the latest developments in at IOR on development of a novel biopolymer based input delivery technique for microbials or maybe other nutrient inputs through seed coating and multi-layer seed coating technologies. We have done some work on groundnut also, so that also I will be sharing. Yeah, my scope of today's presentation is on importance of seed and key factors affecting seed health, seed enhancement techniques and advancements, the crop needs and whatever existing solutions are available, existing technological shortcomings and the scope for advancements, our work at IOR on biopolymers, and multi-crop input delivery through double and multi-layer seed coating techniques. Coming to seed health, I need not to talk much about importance of seed health, and the seed market size is growing day by day. Now, about in the country, about 30,000 crores of seeds are being sold, mostly by private and public sector agencies, and it is expected to grow up to a 50,000 crore mark by 2030. That shows that seed demand is going to enhance, and organized seed production is going to enhance, and there is worries on the importance of these seed health related issues, which is very prime important. Seed is a very important primary input for any success of agriculture and crop productivity. Yeah. Coming to key factors that are affecting seed health, most of you know all these things. These environmental factors during pre and post harvest stages, about to see the factors that affect your seed health, and chemical residues and nutrient deficiencies in the growing plants. All those lead to the seed health, and there are many seed physiological issues at the time of maturity. If there are any imbalances in moisture and moisture stress kind of things, they lead to partial filling of the development of the seed and leads to maybe seed germination related issues. And again, the genetic impurities and look and quality of the materials and weed contaminants. Many times, along with your seed, there are many weed contaminants like your cuscuta and kind of the things they may go along with the seed, causing serious losses during crop growth period. And there are many ways how you just improve your seed health by your pre-harvest treatments, or maybe after during harvest period, or maybe afterwards during processing and storage period also. But if you take some crops like our groundnut and soybean, especially soybean, if you take when it is the harvesting period is usually during October period. Many times it is coinciding with heavy rainfalls. So that is leading to the many issues like plant stress to excess moisture, and then there are foliar pathogens, and there is after harvest and the seed processing, there are a lot of seed contaminants in terms of fungi. They are causing losses during seed germination time. So that is leading to straight away the first season itself. The freshly harvested seed itself is nearly it is losing the viability, and there is nearly 40 to 50% germination only many times we observe. So those are the factors which is the pre-harvest stage issues that is affecting. And you know that during the after harvesting time, there is a damage to your seed, as well as maybe storage period, any moisture imbalance. If there is not required amount of moisture is not there, excess moisture is there, they all lead to attracting some of these storage pests, or many of these storage fungi. So there are many ways by which we can enhance your we call them as seed invigoration techniques. So most of those things you must be knowing, like your hydro priming, treating seeds before a few hours before sowing seed, making seed physiologically active, and then a little bit of drying, and then sowing. And asthma priming by there are many asthmatic protectants like your PEZ, glycerol, and mannitol. That kind of things also can really can do priming, so that some of the some protection as it will be maybe primed, seeds will be primed to tackle some of these issues at the time of asthmatic imbalances and other stresses during germination time, like chemical priming. There are hormonal priming also, and redox priming. So some of these redox compounds, so that the exposing them to a little bit of stress, so that they some of these stress protectants they are accumulated in the seed, so that they can face these any of these stresses like your salinity and the water stress during seed germination period. So there are many techniques like that to really to improve your seed germination and seedling vigor and crop stand establishment in the field. So now today's our topic is mostly on biopriming and biological seed treatments. So this the biologicals in the sense is not only limited to microbials. Maybe there are stimulants and other nutrient mobilizers also for microbial origin, yeah, something like that. So those can be considered for priming, the biopriming. Like yeah, these are the things. So my presentation will be mostly on some of these microbial inputs which can you can utilize to improve seed germination and seedling loss, seedling means the losses during crop establishment period at the seedlings time. Here I have just shown a recent our one of our field on-farm trials, wherein we have seen in a groundnut nearby our in Rangareddy district, in tribal villages. So the seed, there's no seed treatment. How it going to affect your planting stand? You can see on the left hand side you have see there's very means sparse germination and lot of loss due to our collar rot kind of things, and even soil itself there's a decay of seeds because of continuous cultivation of groundnut in that sandy soil type of area for continuously for over many years. So wherein our kind of inputs, the seed inputs are very important. So just a minute. Yeah, currently what may crop needs and existing solution if you take any crop like groundnut, if you take also or soybean, we see the package of practice. There are many recommendations which have to go onto the seed. Like if you take there is an for nitrogen mobilization, there is a rhizobium recommendation, phosphate, PSB mobilization for soil-borne disease control. There is a trichoderma and there is a fungicide recommendation, and there is a insecticide sucking against insects sucking pests, which are helping transmitting some of these bud necrosis kind of viruses. So there are many treatments we advocate in our package of practices, but in practice, most of you know that how much of this groundnut seed is treated in the country. So I have my own experience here in Telangana region, where most of the farmers during sowing time they go for oil mills and they buy seed. groundnut seed. So anyhow, there is no the organized production and supply of the seed to the farmers because high volume crop and no required 60 to 80 kilo of seed requirement per acre. So there's many issues like that. So hardly a few farmers they are going for seed treatment with a fungicide, but many of these inputs are not given. But and expecting farmer to treat at his sowing time also, we cannot expect that a farmer will get all these things at his doorsteps that he can practice all these things. So, but now what are the solutions available really to for the seed treatment? There are single input based products are available in the market for soil-borne disease control. There are many microbials like trichoderma and bacterial biocontrol agents, and there are many nutrient mobilizers. So, many nutrients there are mobilizers nowadays. So there are a lot of research is going on in the country on nutrient mobilizers also, and effective strains are available across many of these laboratories. There are many like your phosphate mobilizing and zinc solubilizers and many of these things. And there are solutions in fungicides also. CT treatment, they're like the most regularly treated chemical is Thiram in many crops. And for nematode control also, there are some of these the Pacillomyces kind of things available for application. And there are a few multi-crop input products also available in the market nowadays. Like many of these companies are coming up with, like your Syngenta, they had a Cruiser Max based on two fungicides and a thimethoxam. And we have BSF product of OTO Prime based on nematicide, that is Bacillus farmas. And same BSF had one more Poncho OTO, that is a systemic insecticide plus Bacillus. These kind of things. Now there are many of these multinational. Earlier, when you talk about microbials, many of these multinationals, majors like your Syngenta, Bayer, or many of these things, they never they look down upon microbials for their production line. But most of these now multinationals they are realize the importance of microbials, let be for plant disease and soil-borne disease management or nutrient mobilization. In the light of the current means these soil health related issues. So many of the soils are better. Their soils are not responding to applied fertilizers. Many of them are getting fixed or immobilized and maybe leaching. There are many issues are happening. So that nearly 50% of any of these fertilizers they're applying, they are not reaching to these means is available to the plant. So and also there's a change in soil reaction. So like your salinity and that kind of things leading to this non-availability of some of these micronutrients also to the plant, to the particular crop. So this is a scenario where the soils are responding to your application of microbials for let be for disease control or maybe nutrient mobilizations. So now this is realized by many of these multinationals, and they are coming with many of these product lines based on microbial agents, apart from some stimulants and other kind of products. Yeah. Yeah, mostly, the current scenario is farmers, if practice, they will go for some chemical fungicides as seed treatment, or maybe very low percentage of farmers have adopted biological solutions. Though we claim that in India there are a lot many microbial producers, let be nutrient mobilizers or maybe disease control organisms. But rarely these are the products which are available to the farmers, and many times farmers won't go for our dealers. They won't means the encourage means supply of these microbial based products to these farmers for seed treatment. So if available also there are single like your chemical may be available locally or maybe one or two maybe nutrient mobilizers or maybe phosphate solubilizers are maybe available, but not the many of these things. Yeah. Most of these products which are available really in the market also, if you take their main problem is their shelf life and quality is anyhow the issue is there, and these strain specificity exists in for many of the crops. Their availability in the soil, short-term protection from pathogens, incompatibility issues. There may not be many times. We don't know if the farmer want to treat a fungicide or maybe chemical insecticide and also by some biological thing. There are no information. There is no information on these their compatibilities and their stability and their viability, and they won't be controlled. Release also is not possible many times, and delivery of bioagents. Most of the products which are available in the market are powder based things or maybe liquid. So what amount of this active ingredient, let be chemical, let be microbe, how much is really reaching onto the seed? So these are issues of seed treatment. Lot of wastage in the pouches or during storage, this treatment time to the vessels. So we won't be sure of how much of real the active ingredient or microbes in terms of quality propagules are reaching out onto the seed. So yeah, to bridge these some of these gaps, like proper delivery and precise delivery of microbial inputs, and also to deliver not only microbial inputs, some of the stimulants and some of the nutrient mobilizers and nutrient inputs also onto the to the seed. So at IOR, we have started work during 2013-14 on delivering these microbials through the available synthetic polymers. Most of you know that most of these market players in the seed market, they go for treating the seeds with synthetic polymer. If you take any one company which is into the commercially producing seeds, if they have turnover is about 100 crores or something, they nearly they spend about 20 to 30 lakhs on buying synthetic polymers. So we started taking that synthetic polymer, try to utilize to deliver some of these microbes. During the process of our research, what you have found, yeah, these are synthetic polymers are they're mostly based on your polyacrylate type of things which are not biodegradable, and their incompatibility with your any of the private companies, they treat seed with some thiram or some insecticides or some one fungicide or one insecticide, and they give. But their synthetic polymers compatibility, if you at all if you want to apply microbes also, their compatibility with this synthetic polymer and synthetic polymer compatibility with other microbes is not clearly known, and they are not also biodegradable also, and they cannot even synthetic polymers may not be able to utilize to deliver multiple inputs onto the seed. Yeah, because the layer capacity will be differing when you are coating onto the seed. So then with and there we found initially we found some issues like that. Some of these synthetic polymers, they are enhancing these storage pathogens, seed pathogens. When you do for go for your germination towel type of testing, when I have seen that in there is a synthetic polymer alone treated seeds, there is an enhancement of this multiplication of more amount of storage seed fungi on the onto the seed. So then we thought that why should we go for this kind of things when there are many of polysaccharides which have already, like you have chitosan and chitin kind of things available, which are really have some antimicrobial kind of thing, and why cannot you convert that kind of these polysaccharides into a means polymeric substances. As such, chitosan is available in liquid form, which form any film around the seed. So it is they use for foliar applications and soil applications and everything. But the thing is that when you put onto the seed, it won't form a uniform film around the seed. So then we have started converting this not only chitosan, cellulose, and some of these xanthan gums and gore gum kind of things, polysaccharides, to whether we can really convert them into a seed coating polymers. So then we have successfully converted these chitosan, cellulose, and some of these gore gums in different combinations, and we have come out with a biopolymer based on cellulose and chitosan, which can form a finer film around the seed, and it went to a combination of materials we have tested, about 700 odd combinations of polysaccharides and cross-linkers, polymerizers, and stabilizers we have tested, and have narrowed down to some 10 or 11 combinations which can form a perfect seed coating around the seed, and as well as it have no effect on the seed germination, which is very important. So that's where you have started our work. We have patented that product also, that biopolymer, and yeah, and you have went on to commercialize that biopolymer recently. Yeah, these are the down. You can see the seeds. All seeds are treated with your these biopolymer in place of your synthetic polymer, and it forms a finer film. You can see on right hand side, there's around circle of film. So, it forms a film like that around the seed. It's not that thick. It forms a fine film, uniform film around the seed. So each film had the capacity to take some amount of active ingredient. So those kind of studies we have taken up. So this is the where we have started testing synthetic polymers and chitosan. We found that synthetic polymer had not that much merit in terms of its the improvement of seed germination or maybe seedling vigor enhancement. And we found that chitosan is imparting that kind of things. So that's where we have started thinking of converting kites onto a polymeric film. So that's finally we were successful in converting this polymeric film. So these are the some studies, initial studies doing 13 and 14, wherein we have found that we need to go for some of the natural polysaccharides to replace our synthetic polymers available in the market. So we have taken up many studies. So these are various combinations of what I am talking about. Various like glycerol, polyethylene glycan, and many of these some of these cross linkers we have tried along with the polysaccharides to convert them into film forming stable film formers. So this is the story which I have shown here about the biopolymer film, how it looks, and biopolymer liquid, and we have a patent on this one. And this technology has been licensed to recently 1 month back to two seed companies at the cost of 25 lakhs each. So there are many publications we have made out of over last 6 to 7 years in high rated journals describing our work, what we have taken up on conversion of some of these natural polysaccharides into polymeric films. So apart from this, we need to, when you want to think of delivering multiple inputs onto the seed, maybe we need to look at maybe organism for each and every problem. So if you have multifunctional strains which can really perform 2 to 3 activities like your plant growth promotion, disease control, and maybe induction responses, and some other foliar pathogens. Maybe sometimes we have a possibility that we can which can ameliorate some of these initial spermosphere salinity issues by absorbing sodium chloride around the germinating seed. So, and there are ways how some of these microbes they can impart or stress or drought tolerances. So, there's such a potent strain, the trichoderma had multiple functions like that. So, at our lab, were able to select a multifunctional strains of trichoderma apart some other microbes which have a very potential means the activity in terms of giving means a biotic and abiotic stress ameliораtions. Yeah. So then after developing our biopolymer, we have started delivering some of these bioagents like trichoderma. We initiated work with the trichoderma, and we were effectively able to deliver through a single layer coating trichoderma. And here we'll have a precision of application. So on each seed, if you can take, we can precisely you can tell about 1,000 to 200 CFE of this trichoderma can be loaded onto the each layer of the on the seed. So that is a precision which is not possible in terms of your if you coat with powders or liquids because there is a lot of wastage and then multiple issues at the time of treatment at farmers level. And shelf life also we are based on this biopolymer based formulation. So the shelf life of trichoderma also is we have taken up to 24 months, wherein we have retained a significant amount of colony forming units also. And each layer of this polymer when coated onto the seed, it takes about it has a entrapment efficiency of 87 to 94%. So like that for each compound, each microbe we can load. It's not that we can load multiple things onto the each layer, single layer itself. It had a limitation. So it loses its integrity and the physical and chemical means other biological properties. When you put too many things, if we add onto the seed, we have to work on entrapment efficiency and the amount of load you can really can a particular layer can take. And accordingly, you need to work how many inputs you have to give onto the each load. So that kind of work you have taken up in the latest slides. I will be showing those things. But this is a story about this trichoderma in a biopolymer based formulation. There's 1% liquid formulation which we have developed and we have tested it in groundnut against this kola rot in groundnut and soybean root rot, macropomena, and safflower also. Some of our oilseed crops we have tested that we have taken to limited field studies as well as on-farm trials also. In safflower also we have taken up a lot many studies in our Eclipse centers. For 2 years we have taken up, and there is a very good improvement in seed germination and plant stand and these productivity enhancement also. In soybean also in soybean network we have taken up these trials in 4 locations. Amravati, Jabalpur, Dharwad, and Adilabad, and we have got mixed kind of results these places wherein there is clearly in some places there is an improvement of seed germination and plant stand and yield improvements. In groundnut also, in locally we have taken up in last few years. We have been doing it in our on-farm trials around in Telangana. So apart from our own research farms, we have taken up and have demonstrated the potential of this biopolymer-based trichoderma. So this in our saffron equip, we have there is a recommendation after in at over 3 years of testing at 5 locations. We have recommended this technology, that is biopolymer based trichoderma harzianum. So 10 ml per kg, or maybe these ever goal extent. So they can be utilized for effective management of soil-borne diseases. Coming to this single input delivery, I spoke till now. So now because any crop, as I said earlier, it is not that it is affected by single means agent or issues. There are many multiple issues during seed germination that particular crop, particular seed it faces. So it may be maybe a drought or a moisture kind of things, and nutrient stresses, and salinity is one main thing, and then there are multiple number of pathogens which they cause seed rotting in the seed in the soil itself before they emerge. And yeah, so when you want to go for direct seed with multiple inputs, there are many issues. You know all those things. Main thing is physical incompatibility, and is leading to many of these inputs which are applied loosely onto the soil and the seed. They may lead to some kind of pollution, change in chemical characteristics. When there is a two inputs are reacting together, there is a incompatible things, there will be adverse chemical reactions also, and antagonistic interactions sometimes when there is incompatible things are applying to onto the seed at the time of sowing. So there is a lot of antagonistic effects of your employed or microbe and maybe any other chemical. Improper adherence, it is known, and many talks it, I think those things I spoke. But this is to drive our point that a seed requires multiple inputs. How much of these inputs you are able to deliver? When the soils are responsive nowadays because of change in soil reaction, and there is a I mean, most of the applied chemical nutrients are mobilized in the soil. They need some means nutrient solubilizers, and they are yeah, that kind of things. And crop demands now multiple inputs, but there's rarely they are available in the market or at farmers' doorsteps that they can go for practicing these things. So by multi-layer seed coating with the different inputs, we can go for means, some these delivery of multiple inputs, and even sometimes incompatible combinations of microbes or chemicals and microbes or nutrients can be combined here. So because there won't be any chemical and physical interaction between two layers, there we can go for a precision application also. So you will be knowing how much of the input you are giving onto the each layer of the seed, and there's proper adherence. There won't be any loss during thing, and there sometimes there's a control release also based on the polymer biopolymer use. There is a release, a control release of your inputs also during seed germination and crop seedling establishment period. So we had to multiple input delivery. We started working on compatibility of some of these biological control agents, pesticides and biofertilizers and these biopolymers. So many combinations we have tried, fungicides, insecticides, and polymers, and finally we found that one chemical which is versatile, that is penflufen trifloxystrobin, is highly compatible with your trichoderma strains, which about three trichoderma strains we have tested with this fungicide. And in biofertilizer, all biofertilizer, this bradyrhizobium, these arachides and japonicum, and bacillus subtilis, all these things are compatible with your fungicide. And also thimethoxam insecticide, we tested two insecticides, and among them thimethoxam is highly compatible with your all these microbial inputs. And anyhow, these polymers, two polymers, biopolymer films, what we have developed are both are compatible. So after identifying these combinations, we went on to work on their delivery, and how much of this each active ingredient we can deliver onto the seed. Yeah, you can see that on the right side of the can photograph with the groundnut treated. So without the polymer, you can see how this is a discontinuous coating onto the seed, and whereas with the polymer, wherein you have done a multi-layer seed coating, wherein is uniform coating around the seed, where there are multiple inputs we have introduced through double layer seed coating. And this is a concomitant. You can see here down panel wherein groundnut in as well as nicer treated seeds. There's a total loss of seed germination and And whereas in combination of fungicide and we have given a double layer coating, a fungicide plus bradyrhizobium and ciliate 2, we have given a cellulose and insecticide and trichodoma. So like that, we have optimized the specific inputs to go with a particular layer and particular combination. So we are like that. We have developed a double layer seed coating in case of groundnut, wherein we have seen clearly. So there is the enhancement of nodulation wherever we have given a bradyrhizobium, and there is a reduction in some of these seed rotting and then color rot incidents while emergence. And yeah, this one you can see here. The talking there won't be any physical interaction after seed coating onto the seed. There are two different layers formed around the seed. You can see this is the first layer having trichoderma and a fungicide, compatible fungicide, and there is a clear-cut demarcation. You can see in the second layer we have a rhizobium onto the seed. These rhizobium cells are visible in the second layer. You can see where we have a insecticide and rhizobium. So there is no physical interaction after seed treatment. These two separate layers are clearly formed. So these things I have already spoke about. Yeah, enhancement of shelf life, and these are the things which we can happen with multi input delivery. Um, yes, I mean, as I told you, even incompatible combinations also you can attempt here, and targeted release of precise quantity of crop inputs. Yeah, persistence also increase the most of the bioagents they can survive more period. Shelf life after even after coating seeds also we have been regularly drawing samples at various storage periods, and we are looking at these seed germination and amount of inputs release from the seed, let be microbes or maybe chemicals. We have been regularly testing from after sowing the pots, how much of these the ingredients are getting released into the soil over a period of time. That kind of tests we have been doing continuously to see that how much of these loaded active ingredients like chemical or microbes are getting released into the soil through different this film coating techniques like your cellulose based film coating and chitosan based film coating. And later on, as I said earlier, that the release of those things we are this employing this entire data to subjecting to different mathematical models, and we have seen that how they are getting released into, after getting released into the soil. It is yeah. In case of your the trichoderma release, you can see from multi-layer seed it follows a non-Fickin type of diffusion. Uh, Here in this non-Fickin kind of diffusion, polymer undergoes swelling, rearrangement of polymer chains occurs gradually, while the diffusion process simultaneously occurs, releasing trichoderma spores entrapped in the matrix. It is a slow release. Yeah, in case of this bradyrhizobium, what you have seen through these mathematical models, everything is that it follows a Fickin diffusion, and therein diffusion rate of solvent transport rate significantly exceeds the thing and facilitating the release of rhizobium in the embedded in the matrix. Yeah, then persistence also. So how long this thing these applying in different layers and individual layers. So how it is getting released trichodoma and how much it is getting proliferated. That kind of data we are taking by in case of groundnut we have taken up in red soils. So yeah. This you can see here in T1, that is multi-layer seed treatment, wherein we have given cellulose with fungicide, and in layer 2 we have given chitosan with insecticide, and layer 3 cellulose and trichoderma. These are layer by layer coating. These are 4 layers, but it is not really, it is not practicable. So that's why we have gone for optimizing the double layer, wherein two inputs we have started giving onto the each seed, wherein we have done chitosan with the bradyrhizobium and trichoderma, and second layer cellulose and cellulose plus this fungicide and insecticide. So this started working very well. So we have seen these good multiplication of our trichodoma and other microbials like rhizobium, which we have introduced under the seed. And apart from the release of the chemicals, whatever you have given, so that you are studying now in the plants as well as in the soil also. So these studies are under progress now. Yeah, even root colonization. So after these sowing with double and multi-layer coated seeds, you are drying samples and looking at the rhizosphere population of your trichoderma and bradyrhizobium also. So there's the improvement of increase in colonies when you are applied in the double layer kind of means applications. So there's significant increase in trichoderma population, and even if you take rhizobium also, the right side we are the graph showing that um, rhizobium population after 60 days of crop growth period after seed application. Yeah. So later on, these double layer and multi-layer coating systems we have started means analyzing in different crops in soybean, in sesame and groundnut we have worked on, and yeah, in soybean we have introduced in two layers. That is chitosan, penflufen, tetrapastrobin, and in layer two chitosan and rhizobium. So there is about 20% improvement seed germination, and there is charcoal rot reduction of 86% over control. This is in germination towel tests and in the laboratory tests. And in greenhouse experiments, we have about 20% and 1% enhancement in germination and about 81% reduction in root rot incidence, so that we can see clearly on the right hand side. So very good means control we get in under laboratory as well as in greenhouse conditions. In sesame also similar kind of observations. There is a study with 29% improvement in seed germination and reduction in seed rot incidence, root rot incidence by macropomena. Similarly, in case of groundnut, we have this sclerotium color. I mean, so this as well as color rot we have taken up study, and there is about 34% improvement in germination and 73% reduction in color rot incidence. That we went on to do some field trials also. Yeah, this you can see in that our field trials on biopolymer technology at ICAR-IIGR form. Your right side panel, seeds which are not treated with any of these input control, you can see there is a sparse germination and lot of gaps you can see. Whereas in right side, is large plots you have done about 4 to 5 plots for each treatment. In double layer coating, you can see clearly this nodulation also. There's very improvement in this number of nodules. A plant stand also you can see is a very good plant stand. And whereas in control, untreated with untreated seed, you can see the amount of plant stand. So straight away there's improvement of seed germination 12 to 13%, disease reduction up to 20% we could see with this kind of multi-input delivery in case of groundnut. Yeah, this is in case of farmers field also. We went on to do this trials in farmers field also. Very good means results we have got in soybean as well as in groundnut also. Yeah, in safflower crop we have validated in our crop locations also by supplying the treated double layer coated seeds to the for FLDs. Yeah. Apart from this, we have further improving upon these our efficiency of some of your applied inputs, like your trichoderma, and by going for development of some of these nano composites based on nano copper, chitosan, and trichoderma based formulations for seed treatment we have developed recently. And this one we are entrapping the biocontrol agent in this kind of matrix like copper, chitosan kind of thing. Will leads to its improvement of bioefficacy. Chitosan many times it serves as a nutrient for its nutrient for some of these microbes, and yeah, so yeah, that kind of biocomposites, nano copper, chitosan based trichoderma biocomposite we have developed. This we have already applied for patent also, and this is a significant improvement of seed germination and reduction of our root and color rot disease in groundnut. So these are very specific examples I'm telling in case of groundnut, but we are doing in our other mandated oil seeds also. Apart from that, many of these, there's a question mark on application of some of the microbes on the foliar environment. They survive well, like your trichoderma, if you take many times, they won't survive for longer periods on the foliar atmosphere, foliar parts because of adverse climate effects or maybe sunlight, and they survival on the unapplied surfaces. So to overcome this one, we have developed a coacervate, now chitosan lignosulfonate based coacervate, wherein it is entrapped in a core of lignosulfonate and chitosan core, where it is slowly released after application on the foliar parts, like let it be on the leaves or maybe on the racemes of any of these or any flowers. So these for application, when this core of means trichoderma with lignosulfonate and chitosan, which is entrapped in that matrix, it will come out when there is a pathogen starts infecting, pathogen starts growing, releasing some of those they started dissolving this matrix of this lignosulfonate matrix. So then it will come in contact with the trichoderma. Till then, trichoderma is in active form. So yeah, in this coacervate. So this we have developed recently. This chitosan lignosulfonate based trichoderma coacervate, which are trying on for their environment. We have initially started doing that in case of control of castor gray mold disease, which is caused by Botulinia ricinae. So you can see on the left side of these plates, you can see the lignosulfonate trichoderma coacervate applied. So there is a little incidence of disease, but whereas in control, there is a excessive, a lot of most of the capsules they got affected by the rotting. So this further we are currently we are working on these experiment part testing of these coacervation of composites, and recently we have completed using these biocomposite based on copper chitosan and this trichoderma for control of macroformina root rot in castor, so that we are now about to complete the trial, and we are getting very good impact means results using this biocomposite. And similarly, in foliar applications, also currently trial is underway in our polyhouse, wherein we are growing castor crop, and now at the resin the capsule formation stage, we are applying trichoderma as well as composites, this coacervate, along with other treatments. Yeah, these are some of the products at IOR which we have developed over a period of time. We have an area of trichoderma based product, trichoderma harzianum 1.5% WP, control of various soil-borne diseases, and we have a high temperature tolerant trichoderma aspergillum strain that is 7316, that's 5% WP, and Trigard, that is a trichoderma harzianum liquid based formulation, that is in SC formulation. This technology has commercialized to seven private agencies, and this biocoat, that is the biopolymer based trichoderma. This we are generating toxicology related information now, as we have all the field bioefficacy testing and everything, and now we are collaborating with some of those private entrepreneurs to further to take this technology forward for seed coating. This is a biopolymer based trichoderma only for seed coating applications, and we have some of novel formulations also. We have is a film based trichoderma. It can be in the form of films only, so that you can, in case of powders and liquids, there's a lot of problem in storage and then transportation costs and everything. And very these films, they are available. Each one film is enough for coating a kilo of seed. So very lightweight, and these films can retain shelf life up to 24 months. Is a novel film. This also we are going to take forward this technology with some collaborative work with some private agencies also. And I spoke already about multi-layer technology and these nano composites and nano these biocomposites also. I have explained a little bit. Coming to this analysis of the biopolymer based biological seed coating. Yeah, there's a lot of interest now in really in say for organic and inputs for natural farming and organic farming kind of things. And many people they realize the importance of beneficial microbes and soil response to these applied microbes, and yeah, their role in this current global climate changes and their requirement of they have the plants they need to face a lot many stresses during stores wherein these microbial inputs they come to their rescue. And there's a technological development wise we need to go for a crop specific customizations. Now we are generalizing everything that we need to we are recommending one means multiple inputs to the multiple crops, and there's no cost crop because as I explained discussed earlier, each crop needs multiple inputs which have to go through the seed also. So when there is a straight away, there's 20 to 30% germination and in seed productivity enhancement, which is nowadays that genetic improvement and the way of yields getting plateaued in many crops. The way you can enhance productivity is by your crop management. Crop management, moreover, your soil health management and delivery of many of these microbial solutions to a particular crop. So, customized crop specific solutions we need to develop So this is my beneficial microbial consortia or something you need to keep working on. There are many challenges of market challenges, adoption barriers, and then the cost effectiveness, and there are many regulatory hurdles. Now itself, there is for single input delivery itself, there are many regulations are there, like your Central Insecticide Board and that our agriculture department level also we have many issues of this marketing and these production license and everything. So you have to overcome when there are multiple inputs you want to give onto the seed. Is possible mainly at processing level itself. Seed processing processor level itself, you can give these multiple inputs. And then farmers will be deriving benefits out of these things. Market opportunities are many. There's a lot of government support nowadays for microbial things. They're supporting many state governments. They are giving some of these microbial inputs by on subsidy also. Coming to future line of these things, you have to, as I earlier also said about customization of crop specific things we need to take up. We have to more and more demonstrations at farmers level and creating awareness, and then when we have to work more on quality of these inputs and scaling up of all these products. So when you want to give a single input self, there is a lot many of these production related issues. When you want to combine many of these microbial inputs and their treatment processes, everything, it requires a lot of means work and energies. Yeah, thank you. This is the one I want to just can talk today. Thank you very much for your patience listening. Thank you.
Thank you, sir. Thank you. A good presentation, sir. And now I request the participants to kindly interact with Prasad, sir. If you have any questions, please feel free to ask.
Dr. Prasad, good evening.
Good evening, sir.
I am Dr. Motilal from Mrudachalam.
Yeah, Pataya, sir.
Have you ever tried the isolation of endophytes from medicinal plants?
Endophytes, a lot of work is going on. Ourselves also, we have started working on this isolating some of the endophytes from our mandated crops also. So yeah, and medicinal plants because it's not our mandate to work on.
Oh, because last week I heard that from Vellore Institute of Technology, they have identified some endophytes isolated from Nerium oleander. So which is highly effective against tomato wilt viruses.
Yeah, that's correct. A lot of work is going on nowadays on endophytes. Now everybody is talking about endophytes and means metabolites. Now more and more work now is going on metabolites of fungal actinomyces or bacterial origin.
Yeah.
So that kinds of things are going on. Yeah, a lot of work is going on in our crops also. In ICAR also, many our research institutes they are working on endophytes. We'll be getting plenty of literature information on that. Yeah, that's all.
Thank you. Thank you.
Good evening, sir.
Yeah.
I am Dr. Narendra from Bikaner.
Yeah, good afternoon.
Very nice talk and presentation on oilseeds, especially on groundnut. You have mentioned, sir, these bioprimers are well working on polar rot also.
Yes.
It controlling around 60% disease as compared.
Polymer. It's not only polymer. Polymer.
Along with the fungicide.
In place of synthetic polymer, we are using a biopolymer. Biopolymer, it had its own inherent merit of antimicrobial nature and defense induction capabilities. You know, chitosan is a very well-known thing. Okay, so that chitosan we have converted into a biopolymer film. Okay, so that we are utilizing to deliver multiple inputs onto the seed. Okay, so then when you have delivered these trichoderma and insecticide, so we could get control of our
potassium postmortem.
We have to mix the microbes and fungicide in this biomolecules.
Yeah, this is a treatment like any synthetic polymers they utilize in the market for mixing polymer with your fungicide or maybe insecticide and they coat onto the seed. Similar way here, compatible combinations you can utilize in a single layer. You can coat as a one layer and then based on an amount of ingredient also. the known amount of ingredient which a particular film it takes. It's not, you cannot put loads of microbes or loads of these chemical active ingredients onto each layer. So that we have to optimize. So optimize quantities of microbe and a particular chemical we can incorporate and put onto the single layer so that it forms a uniform layer without any discontinuity.
So what is the shelf life of this biomolecule?
A shelf life as such by polymer, it had a shelf life, there is no issue at all. When you deliver it to multiple inputs, we are regularly studying now how long this the applied microbe or chemical is active on the seed. So up to we have 12 months, we have taken up the periodically groundnut seed and soybean seed coated with these inputs, and periodically we are studying they release into the soil, how much of the microbe is active first of all, and how much amount of CFUs are available at the particular storage period, and how much effective is that one biologically, it is effective or not. In chemical, similar way, chemical also, how much of the chemical is getting released into the plant and into the soil. That kind of studies we have been regularly doing. Now, till now, we have about 12 months study we have taken up. Every month we draw samples of these double layer coated seeds and then put in germinations and everything, and in pots also, and see draw the soil samples and plant samples and study their perpetuation and translocation, the kind of things we are doing now, chemicals and microbials.
Sir, okay, sir. How we decide dose of these uh, polymers, sir? As per the weight of
That's
seed?
yeah. Really, there is some issues of which compound goes well with particular polymer. So we have seen that cellulose polymer it goes well with your thimethoxam and your rhizobium. That's very good compatible, and they are returning their original population, and there is a effective release of these compounds into the soil as well as into the this plant. So like that, we have studied which are go well with particular combination of like your chitosan. We have found that your fungicide as well as the trichoderma goes well with this particular layer of chitosan. So like that you can do, and many times you can go for the single type of polymer also. It's not that you have to go for each layer one kind of thing. There is minor variations will be there. So yeah, you can adapt maybe one polymer also.
In a multi-layer seed coating, how many layer we can do for either for nutrient, fungicide, insecticide, or
That's how we have done. Not till now, I told you that 4 layers we have done one after other, and we have done double layer also. Means each layer we are delivering 2 inputs. Okay, I explained earlier also. So like that you are done, but there is no issue of germination issues or any other thing. But we found that double layer with 2 inputs each. So you cannot put 3 or 4 or more amount also. So many times it won't, that layer it won't take. So to
okay.
form, yeah, so take, and we have seen. So we can give. Now we are trying to deliver some critical inputs also now. So we have
Okay.
done with fungicides, insecticides. We have done, and phosphate mobilizers and your rhizobium kind of thing and trichoderma, all compatible combinations. Now we are working on delivering of some of the critical inputs, which can be given like maybe micro or major nutrients. Which we are trying to do that also. That part also we are now looking at. So only my thing here is see that the many inputs, whichever particular crop requires goes by doing that one. But is it possible also at in place of seed processing and seed stage, if you can take public sector seed corporations or they involve in this kind of thing. So it's possible that we can give a multiple inputs so that farmers will get benefited. So otherwise, it's very difficult. Farmers they never get this kind of things all together in a place that the farmers apply all these things onto the seed. But these are the necessities now. Now we have to give protect your seed like a delicate seed like groundnut, soybean, so many issues. So you need to protect and you need to give all critical inputs or good inputs, whichever are required for establish good plant stand establishment.
Thank you.
Sir, namaste, sir. This is Vemana, sir.
Yeah, good afternoon, Nagar. Chapani.
Vemana, sir. Vemana, sir.
Yeah,
Sir,
sir. Bataya, sir.
sir.
Tell. How are
Sir.
you?
Fine, sir. Fine, sir. Sir, how much cost, approximate cost required, sir, for 1 acre?
The
by this?
polymer is not that costly. This we are at a market. The synthetic polymers are available at the rate of starting from 400 to 500. So this also it will be in the same range of this thing. Okay, biopolymers also in the now we have fixed price also we fixed around 700 per liter. But anyhow, we cannot give a good technology at a cheaper price to anybody. So that we have fixed at 700 rupees. But otherwise, the production cost won't cross 200 to 250. It depends upon the kind of the quality of chitin, chitosan kind of things we are utilizing. As for agricultural applications, you need to have a all very good analytical grade of these inputs now while commercial scale production. And already some of our two buyers of technology, they started establishing their production units now, and they came out with some of these brand names also. I didn't show those slides. Recently, they have released their products also. One person he has named it as a Biocode, Ecomer by our Hyderabad-based seed company, and other company from Calcutta. So that is a seed primer. So these brands they are available, and maybe by the end of this month they will be in the market also. These two polymers, and they won't be much costlier. Means cost involved in this production. And the only critical thing is how much of these polymerizers and cross linkers you need to use for converting into a polymer film reactor. So yeah.
Sir, it is effective in all types of soil, sir. Especially
We are doing
area, sandy.
yeah, we are doing in black and red soils. We are doing, and this polymers, this biopolymers like your chitin, and they are stable up to 8.5 pH. There is no issue till 8.5 pH, up to probably 9 also, but 8.5, there is no issue for these, especially chitin based, chitosan based biopolymer. There is no issue in the soils with up to 8 to 8.5 pH also.
So I think it is more useful in dryland, sir. Dryland agriculture.
Yeah, dryland. Yeah, that's what now because
Anantapur area in AP, where we must.
Yeah, there is some issue or audio, but anyhow, this is yeah, there is a possibility, sir. With this polymer coating, it will help in during stress conditions also, where there is moisture limiting conditions also. It won't allow it to absorb. Means the limited allow seed to germinate. If the limited moisture is there, it won't. It retains the moisture and it won't allow seed to take. And optimum moisture conditions itself, the seed it will allow the seed to imbibe. So that kind of production and physical production also around the seed, and these chitosan film, it had the antimicrobial nature also. On its own, it had some kind of protection against soil-borne pathogens.
Okay, sir. Thank you, sir. Thank you.
And Dr. Saab, this is Prasad from IIGR.
Yeah, good afternoon, sir.
Good afternoon. Dr. Saab, thank you very much for your understanding and the lecture. Dr. Saab, we have a problem for particularly seed treatment, particularly priming of the seed treatment and mechanical planting. So mechanical planting, what is happen when you go for a priming and you dry upon maybe 2 to 3 hours, so there is a possibility that the embryo get lost or it is damaged. So is there anything that you suggest instead of priming, dry priming or insert of a polymer which will help us for a mechanical planting of a groundnut?
This in case of soybean, you have tested. That is a very delicate seed and everything. There is no issue at all. So you can at the farmer's level also you can utilize the polymer coating seeds by manually. And many of the seed companies, that they have a seed means the production chain wherein they coat seed and there's a little bit of drying it happens, and seed comes out of that the production line. So we can coat. So there is no any issue in the case of soybean. You have tested very well. And in groundnut case also, we have initial doubts that whether it will may damage that, or maybe this aileron layer, it may be peeled off. So nothing happened. We have that is a doubt initially. We have nearly 100, 120 kilos we have treated, and there is no effect. And manually, it's not that we have utilized any of these coating machines. At in a village level itself, we have our people have went and we have coated there and we have sown. So there is no issue at all, sir. In the case of damaged seed, or even it can protect also. Maybe if minor damage, anything is there during the transit period, or in the sachets. So it may offer protection also when a physical layer is there around the seed. It may protect the seed also. But as such, we are periodically drying the samples of soybean and groundnut and other oil seed crops and looking for its germination and other things. There's no reduction in any germination or any other physical issues.
Doctor, besides germination, vigor, and of course pathological reactions, does it increases yield?
Certainly, sir. Yeah, certainly, sir.
10% increase in yield is there. Do you report that this their
Yeah.
yield increase in crop?
Yeah, we have trials. We have trials with polymer alone coated trees also. There is improvement and seedling vigor, though not comparable with other all inputs are given together, but merit or control There is improvement will be there certainly. As such, polymer itself is that's how you claim it's a natural polymer, it's a biopolymer. So which had a potential to replace most of the synthetic polymers in the market, which is a huge market. So, but anyhow, we have to means there are many
So
now.
then, sir, another query is: do you advocate hydrogel versus your biopolymer or polymer? Because the hydrogel people are telling, I have my own experience. Hydrogel does not even beyond 5 kilo also it has no response.
Yeah, that's correct, sir. That's
so,
correct.
what is your personal feeling or experimental feeling, scientific evidences? Is hydrogel will work on particularly semi-arid dry conditions?
Hydrogel, anyhow, our experiments also shows that not much effective in oil seeds.
Yes, sir.
Flower and other things you are tested, but we are not in line in those lines we are working. NASA, this is a biopolymer for seed coating applications alone, and in the places like where there is stresses like seed and soil-borne pathogens, and there's some that kind of things. We are just want to deliver whichever a particular crop is needed. We are not looking at the really moisture kind of things, whether it's a particular certain amount of production it can offer around the seed.
Exactly. Now they are recommending, sir, they are recommending hydrogel fortified with a certain nutrient to help boost the seedling growth.
Yeah, sir.
So is it true? Do you come across some sort of findings?
Yeah, we have done with the hydrogel, sir, earlier long back, 10, 12 years back, and we have stopped it. But now our own, we have a project of NASF now on working on these biopolymers, whatever I am talking about. Lignin based, cellulose based, and all residue based, agriculture residue based, some of this lignosulfonate material, lignin type of materials we are now converting to polymers for soil applications, like to perform functions like similar to hydrogel kind of things. So the natural, but that hydrogels are based on polyacrylates or that kind of things, not that natural compounds. So it has limitations because many.
Sir, do you have maybe one recommendation for a groundnut? So far seed treatment is concerned, we can take it to the reactive level for higher production, or you can say against pathogenic reactions or or something. Can you recommend only one or your product which we can go for that active trial?
Yes, sir. Now we can go for our biopolymer based on chitin that we are ready to supply, and even there are many means 2 companies they are coming out with the product. And at processing stage, now we have seed hubs. That's what I have been demanding also to recently. Secretary has visited a few months back to us, and the joint secretary also seeds. So I am telling them along with seed hubs, give them some product. So this is very simple technology wherein we can produce on your own. It's not that I have to produce myself or you need to buy from a market. As a partners in the same ICR system, it's a simple technology wherein you can produce very simple minimal inputs at the seed hub level itself, and you can quote the seeds. This is a service which you are going to do for a bigger cause. Really, by giving the otherwise now how many times even NSC recently I asked, they said we recommend rhizobium packets we are giving, and only farmers have to apply at the sowing time. That's what the answer I got from the NSC people who are working at and Telangana and Andhra regions. But that is difficult. Many times they won't go for treatment. If you can seed processing stage itself, you can really give the 1 or 2 inputs, which really they can make a change. So that kind of efforts you have to do it, sir. Some service, a service. And our side, we are ready to share the technology to you to produce at locally at seed hub level, any seed hub level. And critical inputs, you have your own. Your groundnut had your own.
Exactly. Sir, we have also 5 products, and not Boost, not Magic Drought Guard. Do you have any feedback of our products, particularly Drought Guard, Sol Guard, or not Boost, or not Magic?
I have not done anything, sir, now, because usually those products are tested in our place. But anyhow, broadly, any input nutrient mobilizers, the plants are responding nowadays. soils are responding. That is the message we have to take, sir.
Yes, sir. This
Most
one.
of the soils are responding nowadays, and you have good number of mobilizers, and there are some stress protectants. Like in on the kind of stress situation also, we have
Exactly.
many bacillus compound species are available, and even trichodoma also. We have trials in safflower and sunflower, wherein we have imparted stress for a prolonged periods, and they have seen how long that particular crop can withstand with and without trichodoma. At least 5 to 1 week period, there is no wilting has occurred in
No,
case of trichodoma treatments.
that is okay.
And there is uptake of and yeah, many things they happen, sir. In case of salinity and high temperature production, they happen with many of these microbials. So it may be there with your products also. That is not that you have to test each and everyone. Maybe you have
Thank you,
tested.
doctor. Thank you so much. Thank you so much.
Sir, myself, Dr. BDS Nathawat, plant pathologist from ICAR-IIGR. Sir, I want to just the trichoderma. We are using trichoderma in which of FIM. Can
Yes.
the liquid trichoderma liquid formulation is also helpful for the soil-borne disease, just like a collar rot?
Sir, any trichodoma, let be in any form, liquid or powder form, is available. It will be effective. Only thing is, how far the liquid formulation is really out of contamination. Any liquid type of things, they attract many things. And you are saying that FIM, adding with FIM.
Yeah.
FIM is out of a high, means means it is goes, what is that, degradation or maturation of the FIM happens in a high temperature in pits. Okay, during that time, lot of many temperature-loving fungi like Aspergillus fumigators, they take over entire FOM. When you apply trichodoma again mix with that kind of things, it can serve as a diluent for direct application in the field. It's not that you can incubate with FOM so that your trichodoma will multiply. So that it won't happen, sir, many times, because already FOM is rich with many of these high temperature loving fungi, and the trichodoma again it is a big competition there. The FOM it has to multiply means. So
Yeah.
it's like that. So you cannot make FIM is this one totally sterile. So that won't happen. Nah? So it's that you can utilize as a diluent to spread in the soil, spread along the rows, that kind of things. You don't anticipate that trichodoma will multiply in that kind of means composted material. Like if you take neem cake, if you take, there's a unorganized collection of neem fruits from the field. There are a lot of hospitalizer
Right.
will develop in the fruits in the field when they start picking that fruits for developing cakes or maybe powder, NSK powders or those things. So it had a lot of already some other fungus which is really to compete with our trichoderma. So you start to take up that kind of things, sir. So they serve as diluents only.
Thank you, sir. Thank you.
Yeah. So anything, Dr. Nataraj? Can we?
Sir, I have one question regarding once it is treated, how long that seed can be retained? Because groundnut tends to lose the viability very fast.
Yeah, you know that genetic longevity, it will be there inherent to the particular seed. That we cannot do anything. Okay, that according to its genetic thing, it will keep means the longevity may go coming down. But by treating this one, it is not going to affect the longevity of your seed. I told you about 12 months I have studied now. There is a control without any treatment. Also, whatever the percentage of these germination is coming down, it will go down, or maybe a little more untreated ones, because there is some physical protection or maybe other moisture loss and everything may be minimized in case of your polymer coated things, which is not the case with your plain seed where which is not coated. Okay, that kind of minor variations they will happen, but not any adverse things. And even more period, little more period, this seed may be viable.
Regarding white grub and collar
Yes.
rot,
Yeah.
have you tried any chemicals for white grub and collar rot together?
No. Color means white grub. No attempts were made. Anything like your Pacillomyces kind of things. There are many things now which are microbials are there available for grubs also.
Yes, sir.
Your matrisium kind of things. But we have not gone to that one because initially we are really dealing with our mandated crops only. As groundnut and soybean are highly delicate seeds and which are really you can show the results there. It's not that if I go to castor crop and the safflower which are thick coated, hull is very thick, so which they won't attract many of these storage pests, pathogens or anything, and they're hardy also. But yeah, you want to see that really our treatment efficiency. So that's why I've gone initially to just to prove this their effect, polymers effect, or maybe their role in seed germination enhancement and everything in case it's soybean and groundnut.
Director, sir. Director, sir.
Maybe the Prasad is busy with some other thing.
Yeah,
I think.
seems
Yeah,
so.
I can,
Actually, Prasad only suggested your name for inviting for presentation. So we contacted you. And thank you, sir, for accepting the invitation.
It's a very important network where really can benefit of this work can be taken forward.
Yes,
So
sir.
I request many of our known people are there in this entire your network. So kindly make use of this one. So for interest of farmers only. So that no way you can.
That's why your information is very lecture is very informative. We have a lot of information. If you supply one or two your product which can be very useful for a CRIP network involving our groundnut crop, it will be very good. So we will be doing and we will be multiplying, we will be needful. So at least one or two more tested product you can give us so that we can test it at our CRIP level.
My request, Doctor, my request is that I think at our coordinating unit level, I provide polymer. Okay, by polymer I will provide. You can treat maybe because every center we cannot ask them to quote and maybe there will be some variations will be there. At least if possible, a few trials in your network wherever this particular problem is there, aiming at particular problem, you can use this your polymer and quote seeds at your level and supply to them, or maybe you can identify few locations where there is a particular problem is there. That particular solution. See, some are commonly required for everything. Like your rhizobium is required everywhere. Your phosphates always are required. Your trichodoma for your most of the monocrop areas where the groundnut is grown continuously year after year. Certainly there will be seed germination and color rot type of incidence will be there. So it will which are reducing to 15-20% of your planting seedling stand. So in such cases, you can go for your rhizobium and trichoderma, maybe any of other critical input, whatever you feel. So those things you can think of yourself in your network level, and I will provide you.
It should be site specific. It should be site specific. Maybe disease, and besides also phosphorus availability, micronutrient availability. That's are also big questions in many of the tracks.
Yes, sir. Yes, sir. So micronutrients we can give. We are not yet started delivering this micronutrient inputs. That there is a demand from our director also that you have to start delivering some of these critical inputs that we are taking up, sir, now one by one.
Thank you, Doctor.
Thank you, sir. Sir,
sir.
बोलिए. It is over?
Yes, sir.
Yeah, that's over.
Okay. So thank you, Dr. Prasad. It's a nice piece of information and discussion we had. And I urge all my pathologists in the coordination unit and also DGR currently do not have any pathologist. So please make use of it, and if you can redirect your research program on this side, or you can join hand with Dr. Prasad, most welcome. So that will, because it is a very important aspect now currently, because particularly soybean and groundnut, there is a lot of issues on seed rate, germination, plant population, establishment of crop. Probably in the beginning itself, after sowing, we are losing our 25 or 30% production due to damage before germination. So in ACRIP trial, we have seen population in the ACRIP trial, population is somewhere very poor or not maintained properly. So it is very important, and we are also last year onwards, we are giving training, advising farmers for having a good germination, but we have a chemical based mostly, but we can use this polymer based technology, or any universities having their own product, they can use a more better way. So ultimately, to have a good establishment of crop through maintaining the population and securing the production. So thank you, Dr. Prasad. Look forward for good work in the system at your university, ACRIF, and our ACRIF also.
Yes.
And it is open for anybody can join hand, and it is easily available in our ICAR system only. So take use of it, benefit of it. So with this, I thank all scientists from ACRIF and all those who have joined today's discussion and enriching, and particularly thank Dr. Prasad for enriching our knowledge and for further utilization of this knowledge. Thank you.
Yeah, thank you, sir. Thank you very much, Bera sir, and thank you for this opportunity to be part of your network, sharing my thoughts. Thank you, sir. Thank you.
Sir, one request from my end. Can I share this Zoom recording with our YouTube channel? Our
Yeah,
YouTube channel.
you can share. You can share, but if possible, edit.
Okay, thank you, sir.
If possible, edit, because sometimes I'll be mixing things many times. Okay, I may not be clear in some places because I won't give much preparation for this kind of presentation. I'll come and just start delivering something. So kindly, if possible, edit and give so that is
Sure,
some
sir, sure.
information it will go. Okay.
Sure, sir. Sure, sir.
Thank you.
Thank you, sir. Thank you, sir. Thank you, one and all, for joining today's guest lecture series. And we'll share this YouTube link with you all. Thank you, Prasad, sir. Thank you.
Thank you. Thank you. I'm leaving. Thank you very much. Thank you. Thank you very much. Bye-bye.
Bye, sir. So we are closing this session. Thank you.